Hey, welcome. Before we start, if you genuinely find what we talk about here interesting and relaxing, please take a moment to like the video and subscribe, but only if you mean it. I'd rather have 5 people who actually care than 1,000 people scrolling through. So let me know in the comments where you're watching from and what you're doing right now. Are you relaxing, working, trying to fall asleep? There's something special about knowing who's tuning in from around the world. Now take a breath, maybe find somewhere comfortable. Dim the lights if you want. What we're about to explore is one of those rare topics that sounds complex, but actually becomes more beautiful the closer you look. Tonight, we're talking about DNA. Not in a panic-inducing genetics textbook kind of way, but in a genuinely calming way. Because DNA, when you really understand it, is maybe the most reassuring thing in the universe. There's something deeply comforting about the fact that inside every cell of your body right now, there's a molecule containing instructions that somehow turned your parents into you. A molecule so organized, so mathematically perfect, that it's been called the most beautiful structure ever discovered. It's called a double helix, and honestly, that's not an exaggeration. Imagine a twisted ladder, not a crude wooden thing from a child's playground, but a perfectly spiraling ladder where every single rung fits exactly into place, where the mathematics is so precise that it never gets confused about what goes where. That's DNA. The sides of this ladder are made of alternating sugar and phosphate groups, stacked together like the rails of an actual ladder. These sugar molecules are called deoxyribose, and they bond with phosphate groups in a predictable, regular pattern that repeats endlessly along the length of the molecule. That's where the name comes from, actually. Deoxyribose nucleic acid, DNA. But here's where it gets genuinely elegant. Each rung of the ladder is made of 2 nitrogen bases, and these bases don't randomly pair up. They're incredibly selective. Adenine always pairs with thymine. Cytosine always pairs with guanine. Always, no exceptions. They're connected by hydrogen bonds, which is like nature's way of saying, I'm holding these together, but not too tightly. These bonds are strong enough to keep the structure intact, but flexible enough to separate when needed. A hydrogen bond is weaker than a covalent bond, which is what holds atoms together within the sugar and phosphate backbone. But in a double helix, there are so many hydrogen bonds, millions of them along the entire length of a DNA molecule, that together they create a bond strong enough to hold the entire structure together through cell division, through copying, through all of life's physical stresses. This complementary pairing system is so reliable, so consistent, that it becomes the foundation for literally all genetic inheritance across every living organism on the planet. Think about that. The same basic pairing system that builds a human also builds a bacterium, a whale, a mosquito, a tree. It's the universal language of life. And what's remarkable is how simple it is. Just 4 letters, A, T, G, and C. With 3 billion of these letters arranged in just the right sequence, you get a human being. Your entire existence encoded in those 4 repeated characters. It's like how our entire alphabet of 26 letters, arranged in different orders, creates all of human literature, music, science, and philosophy. Language encoded in molecules. The human genome, which is all the DNA in your cells combined, contains roughly 3 billion base pairs. That's 3 billion rungs on this molecular ladder. More than 99% of those bases are identical between all humans. The remaining fraction, less than 1%, that's what makes you different from every other person who has ever lived. But here's the genuinely relaxing part. We're so much more alike than we are different. In fact, humans are 99.9% identical in our genetic makeup. Of those 3 billion base pairs, only about 0.5% actually vary between individuals. And while that 0.5% is still what creates visible differences in appearance, personality, and susceptibility to disease, it means we're fundamentally, overwhelmingly the same at our most basic biological level. The genetic differences that make one person tall and another short, one person musical and another mathematical, one person prone to certain illnesses and another resistant, they all come from an incredibly small portion of the total genetic code. Think about that for a moment. Everything that distinguishes you from someone on the other side of the world, every difference in how you look, how you think, how you move through the world, all of that comes from changes in less than 1% of the genetic code. The rest of the 3 billion letters is the same. The same instructions for building bones, the same code for manufacturing enzymes that break down food, the same genetic material for creating the proteins that make up muscles and skin and every organ in your body. This is a beautiful truth. We share a genetic heritage that connects all humans in a biological kinship that transcends any border we've drawn, any language we speak, any culture we've built. At the cellular level, every human being is family. Now, here's something else that adds to this sense of wonder. Every moment of your life, your cells are dividing. Some cells divide slowly, like the cells in your bones, which can take months to fully renew. Some cells divide quickly. Your intestinal lining cells, for example, divide so rapidly that they can renew completely every 4 to 8 days. In fact, you're constantly shedding cells from the lining of your intestines, millions of them every single day. They're replaced by new cells that are being continuously generated, which means your entire intestinal lining is replaced many times each year. And here's the genuinely mind-calming part. Every single time a cell divides, every single time one cell becomes 2, the entire DNA inside that cell copies itself, all 3 billion base pairs. And it does this almost perfectly. The error rate is less than 1 in a million, less than one mistake in a million copied letters. To understand how absurd that is, imagine copying a 3 billion word book perfectly by hand, by yourself, in a few hours, with fewer than 3 mistakes in the entire thing. And then do that over and over again, thousands of times, and never make more than a handful of mistakes. That's what your cells do, constantly, automatically, without thinking about it. That's happening right now, inside your body, while you listen to this. The process is called DNA replication, and it's semi-conservative, which is a fancy way of saying that each new DNA molecule is made of one original strand and one brand new strand. The original strand acts as a template. The new strand is built alongside it, letter by letter, and the pairing rules are so strict that if adenine is on the original strand, thymine is automatically placed opposite it. It's not interpretation. It's not negotiable. It's following the same rule that's been followed for billions of years. Adenine pairs with thymine. Cytosine pairs with guanine. Always. The machinery that does this, proteins called DNA polymerase, have proofreading mechanisms built into them. They check their own work as they go. They're like a typist who reads back what they've written before, moving to the next word. If they make a mistake, they recognize it, back up, and fix it before moving forward. And here's the thing, they're checking constantly. Every single base pair that's added is checked for accuracy. It's not a relaxed standard, it's rigorous. It's not just the machinery though. The cells themselves have backup systems, repair mechanisms that scan through newly copied DNA and look for errors. These are different proteins with different functions, but they're all working toward the same goal. When they find a mistake, they cut it out and replace it with the correct sequence. It's like having both spell check and a human editor, and then having a proofreader look over the work again. It's redundant, it's thorough, it's reliable. It's like the world's most accurate photocopier, but actually better, because it catches and corrects almost every mistake before anyone even notices. The result is that your cells can copy themselves billions and billions of times across your lifetime, and the information stays intact, stays accurate, stays you, billions of times, and the system keeps working. This level of reliability is what makes inherited diseases so rare, and what makes heredity itself possible. If cells couldn't copy their DNA with such precision, every generation would be more broken than the last. Life would fall apart in a few generations. But instead, you look like your parents and your grandparents. You have their eye color because the genes that code for eye pigmentation were copied correctly from them to you. You have your father's sense of humor because the genes involved in personality and psychology were copied correctly. This is not magic. This is chemistry. This is machinery. But the result is that your identity is preserved across time. But here's where it gets almost impossibly vast. If you were to take all the DNA from a single cell in your body and unwind it, if you stretched it out in a straight line, it would be about 6 feet long. 6 feet. In a cell so small you can't see it without a microscope. Imagine that. 6 feet of molecular coil squeezed into a space that's literally invisible. The nucleus of a typical human cell is only about 0.005 millimeters in diameter. That's 5 micrometers. That's so small that 50 of them lined up would just barely span the width of a human hair. And inside that infinitesimal space, there's 6 feet of DNA. But you don't just have one cell. Your body contains roughly 30 trillion cells. 30 trillion. That's 30 million. That's more stars than exist in our galaxy. And that's just how many cells are in your body right now. So if you took all the DNA from all your cells and linked it end to end, it would stretch about 67 billion miles. 67 billion miles. Let me just say that again. 67 billion miles of DNA inside your body. To put that distance into perspective, the average distance from Earth to the Sun is 93 million miles. The distance to Mars is about 50 million miles. The distance to Jupiter is about 484 million miles. The distance to Neptune, the farthest planet in our solar system, is about 2.8 billion miles. The total length of DNA in your body would stretch beyond Neptune and back to the Sun multiple times. Multiple times. You're carrying a DNA molecule of cosmic proportions inside your body every single moment. And the staggering thing is that you're not conscious of it at all. You don't feel the weight of those genetic instructions. You don't sense the cosmic scale of the machinery inside your cells. And yet somehow, incredibly, all of this genetic material is tightly packaged into cells that are invisible to the naked eye. It's a testament to the sophisticated organizational systems that have evolved over billions of years. DNA doesn't just float freely in your cells. It's wrapped around special proteins called histones. These histones are like molecular spools. The DNA coils around these proteins like thread around a spool, making about 1 3/4 turns around each histone protein, forming structures called nucleosomes. These nucleosomes look like beads on a string when viewed through an electron microscope. The nucleosomes cluster together into chromatin. The chromatin further condenses into chromatin fibers. These fibers coil and loop and compress into higher order structures. The chromatin eventually condenses into chromosomes, which are visible under a microscope when a cell is dividing. This layered packaging is magnificent. It's not random. It's hierarchical. It's organized at multiple levels. It's like taking an entire library, all of its books, all of its information, and somehow compressing it into a space smaller than a grain of sand, yet keeping all of it organized and accessible. And not just accessible to be stored, accessible to be read. While the DNA is coiled up around histones, it still needs to be transcribed. it still needs to be copied into RNA, which means the packaging system isn't just about compression, it's about access. Some regions of DNA are tightly wrapped and hard to read. Those tend to be genes that aren't needed right now. Other regions of DNA are loosely wrapped and easy to read. Those are genes that are actively being used. The cell can actually control access to genes by controlling how tightly the DNA is wrapped around histones. It's a physical mechanism of gene regulation. The fact that your body can do this, can store this much information in this little space, keep it organized, and still make it readable when needed, is almost incomprehensible. In the 1870s, a scientist named Friedrich Miescher was studying the nuclei of cells, and he discovered something he called nuclein. He was working with white blood cells from pus-soaked bandages, trying to extract and identify the chemical composition of the cell nucleus. He found a phosphorus-containing acid that was unlike anything he'd encountered before. This was DNA, but nobody realized its significance yet. The chemical was identified, isolated, but its role in life remained mysterious. For decades, DNA just sat on the shelf as an interesting chemical without apparent purpose. Biochemists identified it, understood its basic structure, could measure its molecular weight, but nobody really knew why it was there. Why did cells contain this strange phosphorus-filled acid? What did it do? It seemed inert. It seemed passive. It seemed like maybe just structural material, like scaffolding. In 1943, scientists finally demonstrated that DNA was responsible for heredity, that it was the molecule carrying genetic instructions. It was a transformation in understanding. DNA wasn't scaffolding. It was code. It was the mechanism of inheritance. But knowing what DNA does and understanding how it works are two different things. The structure was still a mystery. How could DNA encode all the information needed to build a living organism? The structure had to contain that information somehow. It had to be organized in a way that made sense. That's what scientists were trying to figure out. How is 3 billion letters organized? What pattern creates meaning? But the real transformation came in 1953. 4 scientists working in the midst of intense competition and collaboration figured out the structure of DNA. James Watson and Francis Crick, working in Cambridge, built models and analyzed data. They were model builders. They believed that if you could construct a physical model that fit all the data, you could understand the underlying structure. They built models from cardboard and wire and metal. They experimented with different configurations. Watson was a young American biologist. Crick was a British physicist. They weren't even the scientists who discovered DNA. They weren't even the ones who ran the main experiments. But they were brilliant at synthesis. They could take data from multiple sources and see the underlying pattern. Maurice Wilkins, in London, studied DNA crystals. He was meticulous. He extracted DNA and coaxed it into crystalline forms that could be studied with X-ray diffraction. And Rosalind Franklin, also in London, took photographs. Franklin was brilliant and exacting. She had a background in chemistry and physical chemistry. She understood crystal structure and diffraction patterns. She was meticulous about technique and data quality. One photograph in particular, taken in 1952, changed everything. It's called Photo 51. It was actually the 51st photograph in a series she was taking, hence the name. It shows a clear X-ray diffraction pattern that unmistakably reveals the helical structure of DNA. The pattern has a distinctive cross shape. The spots arrange themselves in a way that indicates a helix, 2 helices intertwined, visible in the pattern of spots on the photograph. The X-rays are diffracted by the regular repeating structure of the helix. They create spots and patterns that mathematically reflect the underlying molecular architecture. Franklin could see it in her data. The mathematical precision of those spots told her the dimensions, the angles, the exact architecture of the molecule. She was working toward publishing this herself. She was preparing her own paper. Boo, ain't he, before she could publish. Watson and Crick saw the photograph, and they saw immediately what it meant. They understood the implications. Watson and Crick used this data, along with their own models and theoretical insights, to propose the double helix structure. They built a model from metal plates and rods. It showed two strands spiraling around each other, with the base pairs forming the rungs of the ladder. They showed it to colleagues. Everyone immediately recognized that this had to be right. It fit all the data. It explained everything. On April 25th, 1953, they published their findings, a one-page paper. That's it. One page, in the British scientific journal Nature, with the understated title, A Structure for Deoxyribose Nucleic Acid. The paper included a schematic drawing of the double helix made by Crick's wife, Odile. That single page became one of the most influential scientific publications in history. The understatement is almost legendary in science. They proposed the structure of life itself in one page, and they did it with remarkable restraint. They wrote it carefully. They presented the model. They acknowledged the data from other sources. They noted the complementary base pairing. It's beautiful in its precision and its clarity. What's extraordinary about this discovery is that it wasn't one person having a eureka moment. It was multiple brilliant minds working together, each contributing essential pieces of the puzzle. Watson and Crick provided the conceptual framework and model building. They made the intuitive leap. Wilkins contributed precise measurements and crystal structure data. Franklin provided the visual evidence, the photograph that was almost impossible to argue with. The clearest evidence. They worked in a world of intense international competition. There were other scientists pursuing the same question, racing to be first. Pauling, the famous chemist, had also proposed a structure for DNA, but his model was wrong. It had water on the inside, base pairs on the outside. It didn't match the data. But in the end, what mattered was the truth. And the truth required collaboration, even if that collaboration wasn't always acknowledged. Franklin, sadly, never lived to see the full recognition of her work. She died in 1958 of ovarian cancer, just 5 years after the structure was published. And when the Nobel Prize was awarded in 1962 for the discovery of DNA's structure, she was ineligible because the prize is not given posthumously. She had died before the prize could be awarded. Nobel Prizes are typically given 3 to 5 years after the discovery, but Franklin was already gone. Many scientists believe she should have been included, that her contribution was absolutely essential. Watson, Crick, and Wilkins received the prize, but Franklin's legacy was secured anyway. Her photographs, her mathematical analysis, her clarity about what the data meant, that became part of the permanent record of science. She's remembered, if not honored with a prize, then honored with recognition in every biology textbook. The discovery itself represented a shift in how humans understood life. We could now see that inheritance was not some mystical force passed down unchanged from parents. It was chemistry. It was molecules. It was structure and information. The helix explained everything. How you inherited traits from your parents. How mutations could occur. How cells could divide and maintain their identity. How life itself could persist and change across time. The double helix became the symbol of life itself. It was elegant. It was simple enough to explain, but complex enough to encode infinite variety. It was beautiful, and it was correct. The double helix is often called beautiful, and that's not casual language. When you look at the structure, when you really understand the geometry, there's an aesthetic perfection to it. The spiral rises predictably. Each turn of the helix takes about 3.4 nanometers. The pitch, the vertical distance covered by one complete turn, is always the same. 34 nanometers divided by 10 base pairs per turn. The mathematics is perfect. The diameter of the helix is about 2 nanometers. Everything fits. The larger purines, adenine and guanine, pair with the smaller pyrimidines, thymine and cytosine. This ensures that the double helix maintains a uniform width throughout its entire length. If you paired purine with purine, the helix would bulge. If you paired pyrimidine with pyrimidine, it would pinch. But purine with pyrimidine always produces the same width. It's not an accident. It's a direct consequence of the chemical structure of these molecules. Nature didn't design this. Evolution didn't plan it. But natural selection favored configurations that worked. And this one works perfectly. The twist of the helix is about 36 degrees per base pair, which means one complete rotation takes 10 base pairs. This right-handed spiral, this clockwise twist as you move down the strand, this became the canonical image of life itself. Logos of companies, tattoos on scientists, representations of genetics and biology everywhere. It's the double helix. And yet, the structure is even more elegant than most people realize. The major groove and the minor groove, the spaces between the two strands, these allow proteins to recognize and read the genetic code without unwinding the entire molecule. Transcription factors can slide into the grooves, read the sequence of bases, and determine whether to turn a gene on or off. The structure enables function. It's not just beautiful, it's beautifully practical. When DNA replicates, the helix unwinds. The hydrogen bonds between base pairs break, which sounds dramatic but is actually gentle. Hydrogen bonds are weak individually, but they work in pairs or groups, so the separation is manageable. The two strands separate like a zipper unzipping. As they separate, the machinery of the cell moves in. DNA polymerase enzymes attach to each original strand and begin building a complementary strand. On one original strand, a new complementary strand is synthesized. On the other original strand, a new complementary strand is synthesized. The process is continuous but orderly. It's not chaotic. It's not random. It follows rules. The result is two complete DNA molecules where there was one before. Each new molecule consists of one original strand and one newly synthesized strand. This is called semi-conservative replication because the old is conserved, half in each new copy. This mechanism explains how cells can divide while maintaining genetic identity. It explains how you can grow from a single cell into trillions of cells, and every one of those cells, with rare exceptions, contains an exact copy of your complete genome. The precision of this process is staggering. The error-checking mechanisms, called proofreading, reduce the error rate to about 1 mistake per 10 billion base pairs. 10 billion. That means your cells are so careful about copying your DNA that they'd copy a book the size of the Bible thousands of times without making a single error. And even if an error does slip through, there are repair mechanisms that scan the DNA constantly, looking for problems and fixing them before they become mutations. But here's where it gets genuinely strange and contradictory. Not all DNA is genes. In fact, less than 2% of your DNA actually codes for proteins. Less than 2%. That means about 98% of your DNA is not genes. It used to be called junk DNA, dark DNA, non-coding DNA. Scientists looked at these vast stretches of genetic material and thought, what is this for? Why is it here? It seemed like evolutionary garbage, like leftover code that nature couldn't be bothered to clean up. But in the last 2 decades, That understanding has completely changed. Scientists discovered that much of this non-coding DNA plays a crucial role in controlling genes. It acts as a regulatory region. It tells genes when to turn on and off. It protects the genetic code from damage. It stabilizes the structure of chromosomes. It contains information about how cells should develop and specialize. There are regulatory sequences called promoters that sit near genes and control when transcription begins. There are enhancers, which can be located far from the genes they regulate, sometimes hundreds of thousands of base pairs away, and they increase gene expression. There are silencers that ease it. There are insulator sequences that prevent crosstalk between genes. The genome is far more sophisticated than anyone initially realized. The junk DNA is not junk. It's intricate control circuitry. There's an entire field now called epigenetics that studies how genes are regulated, how the same DNA can produce completely different results depending on which genes are turned on and which are turned off. A liver cell and a neuron contain the same DNA. They're both descendants of the same fertilized egg, but they express completely different genes. The liver cell expresses genes related to liver function, detoxification, protein synthesis. The neuron expresses genes for neurotransmitters, ion channels, structural proteins that form connections with other neurons. The difference is not in the DNA itself, it's in which genes are active. And about half of the human genome appears to be made up of mobile genetic elements. These are DNA sequences that have the ability to copy themselves and insert themselves into new locations within the genome. Some scientists call them selfish DNA because they appear to prioritize their own replication over the benefit of the organism. Many of these elements originated as viruses. Millions of years ago, viruses infected our ancestors and integrated their genetic code into the genome. Over time, those viral sequences mutated and changed, but they remained in our DNA. They kept making copies of themselves, inserting these copies throughout the genome. The result is large repetitive regions of DNA that look like they're just duplicating themselves endlessly. And yet, evolution has co-opted some of these selfish elements for useful functions. Some have become part of regulatory regions. Some have been incorporated into genes. Some help control how chromosomes function. It's strange and wonderful, the idea that our genome contains remnants of ancient viral infections, that we're literally made partly of modified virus DNA, that what was once an invader has become integral to who we are. Barbara McClintock discovered these mobile genetic elements in corn in the 1952, and her work was so important that she won the Nobel Prize in 1983. She showed that genetics was not static, that the genome was not a fixed blueprint, but a dynamic, active system where pieces could move around and change. And here's what's genuinely calming about understanding this. It means your DNA is not just a rigid instruction manual passed down unchanged from your parents. It's a living archive. It contains the history of your species. It contains evidence of ancient infections, environmental pressures, evolutionary adaptations. Every feature in every organism was initially the result of a mutation, a change in the DNA sequence. That new variant either helped the organism survive and reproduce, in which case it spread, or it hindered survival, in which case it disappeared. Differential reproduction is the engine of evolution, and mutations are the raw material. While we often think of mutations as harmful, and some are, they're actually the source of all genetic variation. They're the mechanism that has generated the stunning diversity of life on Earth. The calming perspective here is that the very process that creates disease and suffering is the same process that has created beauty, intelligence, complexity, and all the diversity of life. Evolution is always working, always testing, always discovering what works in each environment. In a region where malaria is endemic, a mutation that provides resistance to malaria might be beneficial even if it causes a disease like sickle cell anemia when inherited from both parents. In regions without malaria, that same mutation provides no benefit. But in regions with malaria, having one copy of the mutation is protective. People carrying one copy of the sickle cell mutation have a survival advantage. This demonstrates that evolution doesn't create perfect genes. It creates genes that help organisms survive in their particular environment. Trade-offs are everywhere in evolution. Every advantage comes with a cost. Every change involves a choice. The mutation that causes sickle cell disease has been maintained in human populations because in certain contexts, the benefit of malaria resistance outweighs the cost of the disease. Bacteria exposed to antibiotics sometimes develop mutations that allow them to survive the drug. These resistant bacteria reproduce faster than susceptible ones because the susceptible ones die. Within days or weeks, the population shifts. What was a deadly poison becomes ineffective. Evolution in real time. Beneficial mutations spreading rapidly through populations when the environment selects for them. DNA doesn't just exist in human bodies. It's in every living organism. The more closely related organisms are, the more similar their DNA. Humans and chimpanzees share about 99% of their DNA. We diverged from a common ancestor only about 6 million years ago. Humans and chickens share about 60% of their DNA. Humans and fruit flies share about 60% as well. And humans and cabbage share about 50% of their DNA. 50%. Half of the genetic code that builds a cabbage is similar enough to your own that scientists can recognize it. All life having its origins in the same primordial soup, the same ancestral organism, billions of years ago. This is perhaps one of the most profoundly unifying scientific facts. Every living thing on Earth shares a genetic heritage with every other living thing. The DNA that makes you human also makes cabbages grow. It makes birds sing. It makes fish swim, and octopuses think, and plants photosynthesize, and bacteria divide. We are all made of the same biological building blocks as creatures that seem completely different from us. This shared genetic code means that we're literally related to all of life. That connection, that kinship extending across all of Earth's diversity is beautifully calming. It reminds us that despite apparent differences, all life is fundamentally one family. But understanding that we're all connected genetically is only the beginning. The real beauty of DNA emerges when you start to grasp how it organizes itself, because that organization is nothing short of miraculous. Your DNA doesn't just exist as a loose strand floating around inside your cell. It's packaged, compressed, arranged into a system so sophisticated that it makes most of our technology look crude by comparison. The cell nucleus is already impossibly small. We're talking about a space that's only about 5 micrometers across. That's 5 millionths of a meter. But inside that infinitesimal sphere sits 6 feet of DNA from a single cell. How does that fit? The answer is packaging. Imagine if you had to compress a 6-foot rope into a space the size of a period on a printed page. You couldn't just coil it loosely. You'd need a system. A hierarchy of coiling and folding and compression that works at multiple levels simultaneously. That system exists, and it's called chromatin. The DNA wraps around proteins called histones. These histones are like molecular spools. The DNA makes 1 3/4 complete, turns around each histone protein, creating structures called nucleosomes. When you look at nucleosomes under an electron microscope, They look exactly like beads on a string. One histone protein acts as the bead. The DNA is the string connecting them. This is the first level of packaging. The nucleosomes then cluster together and compress further. They form chromatin. The chromatin fiber coils and loops and compresses into higher order structures. Those structures compress again into even more densely packed arrangements. Eventually, everything condenses into chromosomes, the visible structures you can see and hair a microscope when a cell is dividing. This layered packaging system is magnificent because it's not just about compression, it's about access. Some regions of DNA are wrapped tightly around histones, compressed and hard to read. Those tend to be genes that aren't needed right now. Other regions of DNA are loosely wrapped, easy to access. Those are genes that are actively being used. The cell has learned to control which genes can be read by controlling how tightly the DNA is wrapped around histones. It's a physical mechanism of gene regulation. The system is so elegant that it allows your cells to store information at one density for archival purposes and unfold it when needed for active use. It's not permanent. The wrapping and unwrapping is constantly happening, responding to what the cell needs at any given moment. Think about that for a moment. You have 30 trillion cells in your body. Each one contains the same complete set of DNA instructions, yet they function completely differently. A liver cell does something entirely different from a neuron. A skin cell behaves nothing like a muscle cell. They're not different because they have different DNA. They're different because different genes are turned on and turned off. The liver cell expresses genes related to liver function, making proteins that break down toxins and produce bile. The neuron expresses genes for neurotransmitters and ion channels, creating the electrical and chemical machinery of thought. Same DNA, completely different outcomes. This is the real power of the architecture. The structure isn't just about storage, it's about control. The DNA can exist in a readable or unreadable state based on how it's packaged. Genes can be turned on or off by changing the physical arrangement of chromatin. A region that's tightly packed and inaccessible is essentially silenced. A region that's loosened and exposed is active. This happens dynamically, all the time. In response to what the cell needs, the fact that your body can manage this complexity, can organize 6 feet of DNA in each cell into a system that's simultaneously compressed and readable, organized and flexible, is genuinely remarkable. It's one of those things that seems impossible until you realize it's actually happening right now, inside your body, billions of times over, without you consciously directing it, without you even thinking about it. And the organization doesn't stop at the nucleus. DNA is being read constantly, turned into messages, and those messages are being turned into proteins that go out and do the actual work of living. This is where DNA stops being abstract and becomes visceral. This is where the code becomes flesh. The DNA has done its job. It's stored the information. It's protected it. It's organized it. Now it's time for that information to become action, to become the substances that build your body and keep it running, to become the enzymes that digest your food, the hormones that regulate your mood, the antibodies that fight infection, the contractile proteins that move your muscles. To become you, essentially. Everything that makes you alive is ultimately built from the information encoded in DNA. And the system that translates that information is as sophisticated as the storage system itself. DNA itself doesn't do anything. It's a molecule, a storage device, a library, but the information in it is constantly being accessed, transcribed, translated, and converted into action. The process starts with a molecule called RNA. RNA is like DNA's temporary counterpart. It's similar in structure, almost identical in some ways, but it's single-stranded instead of double-stranded, and it uses a slightly different sugar. The process of creating RNA from DNA is called transcription. It's like making a photocopy of a single page from the library. You don't take the entire book. You copy just the page you need, and then you take that copy and use it. The copy can be made multiple times from the same original. The original stays safe in the nucleus. The copy goes out into the cell and gets used up. This is profoundly practical. DNA is precious. It's the master copy. It needs to be protected. But RNA is temporary, disposable. It can be made and destroyed without consequence. A single gene can be transcribed into dozens of RNA copies. Each copy can be translated into dozens of protein molecules. A single instruction multiplied across many copies, each producing many results. This is how a single gene can have a massive impact on the cell. One piece of information, copied and copied again, amplified into widespread effect. The machinery that does this transcription is a protein called RNA polymerase. It's an enzyme, which means it's a protein that catalyzes reactions, that makes things happen. RNA polymerase is like a copying machine. It reads the DNA sequence and builds a complementary RNA strand. It moves along the DNA, reading one letter at a time, adding the corresponding RNA letter to the growing strand. A at C in DNA becomes U in RNA. G stays G, C becomes G, T becomes A. The process is automatic. The rules are absolute. The enzyme just follows them. But before transcription even begins, the cell needs to know which genes to transcribe, which pages to photocopy. This is where gene regulation comes in. Gene regulation is how a cell controls which genes are expressed out of the thousands of genes in its genome. It's how a liver cell decides to make liver proteins and a neuron decides to make neuron proteins. The answer lies in regulatory regions of DNA called promoters and enhancers. A promoter sits near a gene and acts as an on-off switch. When a promoter is active, the gene it controls gets transcribed. When it's inactive, the gene remains silent. An enhancer is a regulatory sequence that can sit far away from the gene it controls, sometimes hundreds of thousands of base pairs away, and it increases the expression of that gene when activated. A silencer does the opposite, decreasing expression. These regulatory regions are controlled by proteins called transcription factors. Transcription factors are the control panel of the genome. They bind to DNA at specific sequences and either activate or repress gene expression. Some transcription factors are activators, proteins that boost a gene's transcription. Others are repressors that decrease it. The cell produces different transcription factors in response to different signals. A hormone arrives. It binds to a receptor on the cell. The receptor activates a transcription factor. That transcription factor enters the nucleus and binds to an enhancer. The gene is now turned on. A different signal arrives. A different transcription factor is activated. A silencer is engaged. The gene is now turned off. It's exquisitely responsive. It's like a dimmer switch on a light. You're not just turning it on or off. You're controlling the intensity. Different levels of transcription produce different amounts of protein. A liver cell needs a lot of liver enzymes, so the transcription factors for those genes are highly active. The enhancers are open. The chromatin is loosened. The genes are transcribed constantly. A different cell type doesn't need those enzymes, so those same genes stay wrapped up and inaccessible, silent, inactive. The sophistication of this system is almost incomprehensible. Thousands of genes, each with its own regulatory elements, each responding to different signals, each producing different amounts of protein based on the cell's needs. And this is happening in 30 trillion cells simultaneously, each cell responding independently, but in coordination with the whole organism. Once RNA has been transcribed, it leaves the nucleus and enters the cytoplasm, the gel-like substance that fills the cell. But here's something genuinely remarkable. The RNA that leaves the nucleus is not exactly the same RNA that was transcribed. It's been edited, processed, modified in ways that reflect the sophistication of cellular life. The RNA contains information for making a protein, but it's wrapped up with non-coding sequences called introns, interspersed throughout the coding sequences called exons. These introns are like advertisements in a newspaper. They're surrounded by the actual text, but they're not part of the message. So the cell has to remove them. Enzymes called spliceosomes recognize the boundaries between exons and introns and cut them out. The exons are stitched back together. The introns are degraded. The result is mature mRNA, messenger RNA, ready to be translated. This process, called RNA splicing, is remarkably flexible. A single gene can be spliced in different ways, producing different mRNA molecules, different proteins, from the same DNA sequence. This is how genetic diversity emerges without gene-etic mutation. The same gene can have multiple functions depending on how it's spliced. The cell can make different decisions about which exons to keep and which to discard. It's like reading a book where you can skip different chapters and get different stories from the same source material. The mature mRNA is also modified at both ends. A cap is added to the front. A tail is added to the back. These modifications protect the RNA from being degraded too quickly. They signal to the cell that this RNA is important and should be translated. Different lengths of tail can influence how long the mRNA persists and how much protein is made. The regulation is happening at every level, not just at the level of transcription, deciding whether to make the RNA in the first place, but at the level of processing, deciding what kind of RNA to make, deciding how much protein will be produced from each RNA molecule. The cell is constantly making decisions, constantly controlling the flow of information, constantly optimizing what gets made and how much. In the cytoplasm, the mature mRNA encounters another remarkable system. Ribosomes. Ribosomes are protein synthesizing machines. They're not alive. They're not organisms. They're just structures made of RNA and protein that catalyze a specific reaction. The reaction is called translation. Translation is the process of converting the genetic information in RNA into a physical protein. The ribosome reads the RNA sequence, 3 letters at a time. These 3-letter groups are called codons. Each codon specifies a particular amino acid. There are 4 letters in the genetic code: A, U, G, and C. With 3 letters, there are 64 possible combinations, 64 codons, but there are only 20 amino acids used in proteins, so some amino acids are specified by more than one codon. Leucine can be made from 6 different codons, arginine from 6. Some amino acids, like methionine, are specified by only 1. The redundancy is elegant. It means that some mutations in the DNA won't change the protein at all. A mutation that changes the 3rd position of a codon might not change the amino acid that's inserted. This is called the wobble hypothesis, and it's a built-in safety mechanism. The genetic code can tolerate some mutations without consequence. The ribosome pulls amino acids out of the cytoplasm and links them together in the sequence specified by the RNA. One amino acid, then another, building a chain, a polypeptide. Each amino acid is brought to the ribosome by a molecule called transfer RNA, tRNA. The tRNA matches the codon in the mRNA with the correct amino acid in the cytoplasm. It's like a matchmaker. The tRNA recognizes a codon through base pairing, just like DNA base pairing. The codon on the mRNA pairs with the anticodon on the tRNA. It's an elegant system. The same base pairing rules that keep the double helix stable are used throughout the cell to match information with function. Codons match with anticodons. mRNA matches with tRNA. Information is recognized through complementarity, through the principle that A pairs with U in RNA and G pairs with C. The chain grows, amino acid by amino acid. The ribosome moves along the mRNA like a train on a track, reading each codon in sequence, adding the corresponding amino acid to the growing chain. When it reaches a stop codon, 3 letters that signal the end of the gene, the process halts. The completed protein is released. The ribosome lets go. The protein is free. The chain folds into a specific 3-dimensional shape, and that shape determines what the protein does. The 3-dimensional structure of a protein determines its function. The shape creates active sites where chemical reactions can occur. The shape determines whether the protein can cross a membrane or stay inside the cell. The shape determines whether the protein can bind to other proteins or to DNA or to hormones. Everything depends on shape, and shape depends on the sequence of amino acids, and the sequence of amino acids is determined by the sequence of codons in the RNA, and the sequence of codons is determined by the sequence of bases in the DNA. The information flows downstream. DNA encodes RNA. RNA encodes protein. Protein creates function. This is the central dogma of molecular biology. It's the core principle that explains how information becomes action, how instructions become reality, how the microscopic code in your nucleus becomes the living, breathing, thinking organism that is you, and it's happening constantly. Right now, billions of ribosomes in your cells are reading mRNA molecules and assembling proteins. Every second, every moment. Your cells are dying and being replaced. Your proteins are being worn out and rebuilt. Your hair is growing because cells are dividing and proteins are being synthesized. Your immune system is active because specific proteins are being made in response to pathogens. Your heart is beating because proteins in your heart muscle are contracting. The entire operation of your body depends on this flow of information. DNA to RNA to protein. Instructions becoming flesh. Code becoming chemistry, becoming biology, becoming you. The precision of this process is staggering. A single base pair change in the DNA can alter an amino acid in a protein. That alteration can change the protein's function. That change in function can cause a genetic disease. A mutation that changes a single amino acid in the hemoglobin protein causes sickle cell disease. A mutation that changes a single amino acid in a protein involved in nerve growth causes cystic fibrosis. A single letter out of 3 billion. One mistake, and it can change a life. But the beauty is that the system is so reliable that these mistakes are rare, and when they do happen, the cell has repair mechanisms to catch them. Error checking, proofreading, multiple layers of quality control ensuring that the information flows correctly from DNA to RNA to protein. The central dogma is not just a scientific principle, it's the explanation for how life works, how you work, how every organism works. The same principle applies from bacteria to humans. DNA makes RNA. RNA makes protein. Protein does everything else. And here's where it gets truly beautiful. The same DNA is in a liver cell and a neuron. The same RNA can be made from that DNA. The same proteins could be produced. But the neuron doesn't need liver proteins. So the genes for liver proteins aren't transcribed. They remain wrapped up in chromatin, inaccessible, silent. Instead, The neuron produces neuron proteins. The same genetic information, used selectively, produces completely different outcomes. This is the secret of multicellular life. Every cell contains the entire instruction manual, but each cell reads only the pages it needs. This is possible because of gene regulation, because of the transcription factors, because of the enhancers and silencers and promoters that control which genes are read when. It's an exquisitely sophisticated system of control, and it works at every level. At the level of chromatin structure, controlling physical access. At the level of transcription factors, controlling which genes are activated. At the level of RNA stability, controlling how long RNA molecules persist in the cell before being degraded. At the level of protein processing, controlling how proteins are modified after they're synthesized. At the level of protein degradation, controlling how long proteins persist in the cell. The system is resilient. Multiple layers of control ensure that the right genes produce the right proteins in the right amounts at the right times. If one layer fails, others can compensate. The sophistication is astonishing, but it's also reassuring. It means your cells aren't chaotic. They're organized, they're responsive, they're adapting constantly to what you need, automatically, invisibly, without you having to think about it or direct it. But this perfect system, this elegant architecture of regulation and control, is built on a foundation of imperfection. Life itself depends on mistakes. Without mistakes, there would be no variation. Without variation, there would be no evolution. Without evolution, life would be trapped forever in whatever form it first took billions of years ago. Life persists and evolves and adapts because the system that copies DNA is almost, but not quite, perfect. Mutations are errors, changes in the DNA sequence. A base pair that was supposed to be adenine thymine becomes something else. A section of DNA gets duplicated or deleted. A stretch of DNA gets inverted. These are mistakes. These are broken copies. And yet, these mistakes are the raw material of evolution. Every single genetic feature in every organism was initially the result of a mutation that changed the DNA, a mutation that produced a new protein, a new function, a new capability. If that mutation helped the organism survive and reproduce, it persisted. It spread through the population. If it hindered survival, the organism died before reproducing, and the mutation disappeared. This is natural selection. This is evolution. Differential reproduction based on heritable traits. The mutations that persist are the ones that work, the ones that help, the ones that improve survival or reproduction in the current environment. But here's what makes mutations genuinely fascinating. They're not always harmful. In fact, some of the most important features of organisms came from mutations that were beneficial. The mutations that gave wings to birds, that gave opposable thumbs to primates, that gave complex brains to humans, that gave camouflage coloration to insects, that gave venom to snakes. All of these started as mutations, changes in DNA, small changes that, over generations, accumulated into the diversity of life. The process is slow. A single mutation might change a protein's function slightly, but over thousands of generations, over millions of years, small changes accumulate into big ones. Insects that were dark-colored in forests became darker because mutations toward darker coloration helped them hide from predators. Fish that lived in caves lost their eyes because mutations that damaged eye development were neutral in complete darkness. Humans that lived in northern latitudes with less sunlight developed lighter skin because mutations that reduced melanin production allowed more sunlight to be converted to vitamin D. The process is subtle, it's not directed, it's not purposeful, it's just that certain mutations helped in certain environments. And in those environments, organisms carrying those mutations had more offspring. And so the mutations spread. The calming perspective here is that the very process that creates disease and suffering is the same process that has created beauty, intelligence, complexity, and all the diversity of life on Earth. Evolution is not about perfection, it's about adaptation. It's about organisms fitting themselves to their environments over time. And mutations are how that fitting happens. Without them, life would be frozen, stuck, unable to respond to change. A meteor impact would kill everything because evolution couldn't happen fast enough to adapt. A new disease would wipe out entire species because they couldn't evolve resistance. A climate change would cause extinction because organisms couldn't evolve to tolerate new conditions. Mutations are the mechanism that allows life to persist in the face of constant change. They're the safety valve, the backup system, the way forward. Consider the sickle cell mutation. The mutation occurs in the beta-globin gene. A single nucleotide is changed. Adenine becomes thymine. That's it. One letter. But that one letter changes the codon. The codon that specifies glutamic acid is changed to a codon that specifies valine, one amino acid out of 146 in the beta-globin protein. That single change causes the hemoglobin molecules to polymerize under low oxygen conditions. They form long fibers that distort the shape of red blood cells. Normal red blood cells are smooth and flexible, able to squeeze through tiny capillaries. Sickle cells are rigid and crescent-shaped. They get stuck in blood vessels, blocking blood flow, causing pain, tissue damage, organ failure, early death. Having two copies of the sickle cell mutation causes sickle cell disease. Red blood cells become rigid and sickle-shaped. This is harmful. This is suffering. But having one copy of the mutation, carrying it from one parent, the cell contains one normal hemoglobin gene and one mutated one. This provides some protection against malaria. The parasite that causes malaria prefers to infect red blood cells that are completely normal. It struggles in red blood cells that contain even some sickle hemoglobin. So people carrying one copy of the mutation are partially protected. They're less likely to get severe malaria. In regions where malaria is endemic, where mosquitoes carrying the parasite are common, where malaria is a leading cause of death, this protection is valuable. It's the difference between life and death. Life expectancy with sickle cell disease, even with modern medicine, is reduced. But malaria, untreated, kills. So, in the evolutionary calculus of those regions, carrying one copy of the sickle cell mutation increases survival. It increases reproductive success, so the mutation persists in those populations. It's maintained by natural selection because the benefit of malaria resistance outweighs the cost of the disease in carriers. It's a trade-off. You get some protection against malaria, but you get some increased risk of blood vessel complications. On balance, in a malarial environment, that trade-off favors survival. In regions without malaria, the mutation provides no benefit. It's purely harmful. Carriers have a slightly increased risk of complications without any protective advantage, so the mutation is less common there. But in regions with malaria, in West Africa, in the Mediterranean, in the Middle East, in India, you'll find the sickle cell mutation at high frequency. In some populations, 20 or 30% of people carry at least one copy of the mutation because it helps them survive. This is evolution at work, a mutation that creates a trade-off, harmful in one context, beneficial in another. The organism adapts to its environment by carrying mutations that help it survive in that environment. The beautiful thing about this example is that it shows that evolution doesn't create perfection. It creates adaptation. The sickle cell mutation is not good. It's not optimal. But in a specific environment, it helps more than it hurts. Evolution doesn't wait for a perfect solution. It uses whatever works. Or consider antibiotic resistance in bacteria. Bacteria are exposed to antibiotics. The antibiotics kill most of the bacteria. But some bacteria have mutations that make them resistant to the drug. These resistant bacteria survive. They reproduce. They pass the mutation to their offspring. Within days or weeks, the population shifts. The resistant bacteria become common. The non-resistant ones are gone. What was a deadly toxin to the population becomes ineffective because the population evolved resistance. This is evolution happening in real time. Mutation creating variation. Natural selection acting on that variation. The population adapting. It's happening in hospitals right now. Antibiotic-resistant infections are a public health crisis because bacteria are evolving faster than we can develop new antibiotics. It's a race. And the bacteria are winning because they reproduce quickly, they have high mutation rates, and natural selection is ruthless. Any bacteria with a mutation that helps them survive the current antibiotic gets to reproduce. The mutation spreads. The population becomes resistant. Then a new antibiotic is introduced. If there's a mutation for resistance to that antibiotic, it spreads too. The bacteria keep adapting. They keep evolving. It's not malicious. It's not intentional. It's just evolution working exactly as it does everywhere in life. Natural selection favoring mutations that increase survival and reproduction. Every time a mutation occurs, every time a cell divides and makes a copying error, every time DNA is damaged and repaired imperfectly, a variation is introduced into the population. Most of these variations are neutral. They don't help or hurt. They just exist. A different sequence but with the same function. The organism is unaffected. Some variations are harmful. They decrease fitness. They make the organism less likely to survive or reproduce. They're selected against. The organisms carrying them are less likely to pass on those mutations. They disappear. But some, in certain environments, are helpful. They increase fitness. They make the organism more likely to survive or reproduce. They're selected for. The organisms carrying them are more likely to pass on those mutations. The mutations spread through the population. Over generations, they become common. Over millions of years, small changes accumulate. Species adapt. Speciations happen when populations diverge so much that they can no longer interbreed. New forms of life emerge. Entirely new species. This is how life has filled every niche on Earth. This is how the diversity of life has exploded since the first self-replicating molecules appeared billions of years ago. From bacteria to plants to animals to humans, all emerging from a single common ancestor through the accumulation of mutations and natural selection. This is how you came to exist. Your ancestors faced endless challenges: predators, disease, starvation, extreme weather, environmental changes, seasonal shifts, ecological disruptions. And they survived, not because they were perfect, but because they had mutations that helped them adapt to each new challenge. A mutation that made them faster. A mutation that gave them better color vision. A mutation that allowed them to digest a new food source. A mutation that made them more social, more cooperative, more capable of forming complex groups. Every ancestor of yours passed on their genes because they had some trait, coded for by some gene, influenced by some mutation, that helped them survive and reproduce. Every organism alive today is the descendant of survivors. Survivors who had mutations that helped them overcome their environment. And those mutations are still in your DNA, coded into you. making you who you are, your capacity to think, to move, to perceive, to survive. All of it traces back to mutations, to mistakes in copying, to errors that over generations became features, became advantages, became you. This is not just abstract. This is visceral. This is real. The genes that make your brain capable of understanding these words, the genes that make your eyes capable of reading, the genes that make your fingers capable of typing or touching, all of these trace back to mutations, to random changes in DNA that happen to help your ancestors survive. The mutations that killed others created you. The mistakes that failed in other lineages succeeded in yours. You exist because of mutations, because of errors, because the system of copying DNA is almost perfect, but not quite. And that imperfection is everything. You are a library. That is not a metaphor. It is a literal fact. Your cells are not just containers of random chemistry. They are repositories, archives, storage vaults. Every cell in your body carries the complete instruction set for building a human being. Not just any human. You. Your specific instructions. Your unique combination of traits, capabilities, and potentials. 3 billion base pairs. That is not just a number. 3 billion. If you tried to count them, saying one number per second, non-stop, without sleeping, without eating, without pausing, it would take you about 95 years. Nearly a century to count them all. And that is just one copy of your DNA in one cell. Your cells contain billions of these instruction sets simultaneously. 30 trillion cells, each one a library, each one a complete reference copy of the genetic information that makes you you. This is not abstract. This is visceral. Right now, at this very moment, somewhere in your body, a cell is dying. Not in a traumatic sense, not from injury or disease, just normal death. A cell has divided too many times and reached its limit, or it has simply completed its term. A liver cell might function for about 9 months before being replaced. A skin cell from your epidermis lasts only about 4 weeks. A red blood cell in your blood survives for about 4 months. But when these cells die, their replacements are already being made. And those replacements need instructions. They need to know how to be liver cells, how to be skin cells, how to be blood cells. Those instructions are in the DNA, the same DNA sequence that has been in your cells since you were born, since you were conceived actually. A sperm cell carrying half your genetic instructions merged with an egg cell carrying the other half. And in that moment, your complete genetic library was assembled. That library has never changed. Every copy of that DNA made since has been a duplicate of that original library. You carry inside you the same genetic instructions you had at conception, the same 3 billion base pairs. They are in every cell that divides, every cell that replaces its predecessor, every cell that responds to injury or illness. When you grow new skin after a burn, the cells being created are following the original instructions. When your bone breaks and heals, the osteoblasts laying down new calcium are following the original blueprint. When you get an infection and your immune cells multiply to fight the pathogen, they are dividing according to the original genetic code. The library is constant, permanent. It does not change with age, injury, or experience, at least not the DNA sequence itself. What changes is which parts of the library are read, which instructions are being followed, which genes are active, but the books remain the same. Think of it another way. Imagine you inherited a house, not just any house, but one that is designed so perfectly that it repairs itself automatically. When a wall begins to crack, cells in the wall detect the damage and activate repair mechanisms. When the roof starts to leak, the structure sends signals that trigger the production of new roofing materials. When part of the foundation weakens, reinforcements are manufactured and put into place. But here is the crucial part. The house follows the same architectural plans to make all these repairs. The plans never change. Only the specific rooms being worked on change. Only which tools are being used change. But the fundamental design, the blueprint, remains constant. This is your body. Your DNA is the blueprint. It is the permanent record. It is the library that will never be rewritten or fundamentally altered. And yet, that permanent, unchanging library contains instructions for an organism that is constantly changing, constantly adapting, constantly responding to its environment and circumstances. The permanence of the code exists alongside the dynamism of life. The static blueprint produces dynamic results. And somehow, impossibly, this balance works. Your body uses that unchanging library to create all the changes it needs to survive, to grow, to heal, to function. But the library is older than you. Far older. The DNA in your cells right now contains not just instructions for building a body, but a record of history. A very long history. The base pairs in your DNA carry echoes of every ancestor you have. Every parent and grandparent and great-grandparent stretching back through time. You carry DNA that has been passed down through thousands of generations. DNA that survived every challenge those ancestors faced. DNA that was selected for, maintained, and carried forward because it helped your ancestors survive. When you look at the DNA in your cells, you are looking at a record of survival, a chronicle of endurance written in the 4 letters of the genetic alphabet. This is where DNA becomes genuinely awe-inspiring. Not just as a storage system, though it is that, but as a historical record, a compressed archive of millions of years of evolution. The DNA in a human and the DNA in a chimpanzee are 99% identical. We share so much genetic code that the differences are almost negligible, but in that 1% difference, the entire trajectory of divergence is encoded. The mutations that accumulated over millions of years of separate evolution, the changes that made humans human, not dramatic changes, not sudden transformations, tiny alterations, single base pairs that changed, that accumulated, that gradually produced different proteins, different capabilities, different futures. And you can trace those changes. You can look at the DNA of a human and a mouse and see that 85% is identical. A human and a dog, 84%. A human and a fruit fly, about 60%. A human and a zebrafish, 70%. A human and a chicken, 65%. A human and a plant, 50%. The more distantly related the organism, the fewer genetic similarities. But always, there is similarity. Always, there is recognizable overlap. Because all life on Earth shares a common ancestor. And that ancestor's DNA was passed down and modified over billions of years. Every organism alive today carries DNA that traces back to that common root. We're all relatives, all cousins, all sharing the same ancient library, modified by time and evolution, but fundamentally connected. The DNA in your cells carries that connection. It contains the history of your species, the evidence of how humans diverged from our primate ancestors, the genetic changes that led to bipedalism, to complex brains, to language, to consciousness. Those changes are encoded in your DNA, not as complete narratives, but as variations in the sequences, as differences in the base pair arrangements. The history of how apes became humans is written in the genetic differences between modern humans and modern apes. We are reading that history. We can see where the divergences happened. We can identify the genes that changed, the mutations that accumulated, the selective pressures that favored certain variations and disfavored others. But the history extends even further back. Before humans, before apes, before mammals, before vertebrates, before multicellular organisms, all the way back to the first life. The single-celled organisms that emerged from chemical reactions in the primordial oceans. That ancient DNA, modified and complicated and branched and elaborated over 3.5 billion years, is still in you, not unchanged, extensively modified, but fundamentally the same system, the same replication mechanism, the same base pairing rules, the same molecular machinery of life. When scientists look at bacterial DNA and human DNA, they see the same fundamental architecture, the same logic, the same language expressing different stories, but using the same alphabet. This is not spiritualism or poetry, though it feels that way. This is literal biological fact. Your DNA connects you not just to your parents and grandparents, but to every human who has ever lived. To your ancient ancestors from hundreds of thousands of years ago. To humans from 50,000 years ago. To humans from 500,000 years ago. To human-like creatures even further back. To primates. To mammals. To vertebrates. To fish. To invertebrates. To plants. To fungi. To bacteria. You are made of the same genetic material as all of them. You are their descendant. You are their inheritance made flesh. The DNA in your cells is not just your personal library. It is a shared library. A species library. A library of life itself. And that library has been accumulating knowledge. By holding complexity, recording information for longer than human history, longer than human civilization, longer than human beings have existed. But here is the remarkable thing about this ancient DNA. It persists, it survives. For 3.5 billion years, DNA has been copied, modified, damaged, repaired, and passed on. 3.5 billion years. To put that in perspective, the Earth itself is only 4.5 billion years old. DNA has existed for nearly 80% of the Earth's entire history, and it has survived everything. It has survived asteroid impacts that killed most life on Earth, 5 major extinction events, catastrophic climate shifts, ice ages and volcanic winters and atmospheric collapses. It has survived in organisms that barely survived themselves. Organisms reduced to tiny populations. Organisms pushed to the edge of extinction and clawing their way back. And yet the DNA persists. It always persists. Why does DNA persist? Partly because it is durable. The molecule itself is remarkably stable. The double helix structure, with the bases protected inside and the sugar phosphate backbone on the outside, makes the DNA relatively resistant to damage. Not invulnerable, but robust. More durable than RNA. More stable than protein. DNA is built to last. Built to be copied. Built to be preserved. The structure itself is a form of redundancy. You have 2 copies of the information, one on each strand. If one strand gets damaged, the other can be used as a template to repair it. The cell has evolved sophisticated systems specifically for reading one strand and using it to fix damage in the other. It is like having a backup copy of every document. If one gets wet or torn, you have the other to rely on. And yet durability alone does not explain persistence. DNA persists because organisms that carry it have survived. Natural selection has favored DNA-based life because DNA is an efficient information storage system. It replicates accurately. It stores information in a way that can be read and translated into function. It can be repaired when damaged. It can be modified by mutation without becoming completely non-functional. It is a system that works. It works because of the way it is structured. The redundancy built into the system means that mutations often do not cause catastrophic failure. A change in the third position of a codon often does not change the amino acid. A mutation that affects one strand might be fixed by the error correction machinery before it becomes a problem. A damaged gene might be backed up by a regulatory mechanism that shifts function to another gene. The system is resilient. It has built-in safeguards, multiple layers of protection, and those protections exist because organisms that had better error correction, better repair mechanisms, better ways of dealing with damage, left more offspring. They outcompeted organisms with less robust systems. Evolution selected for DNA stability, not perfect stability. If DNA were perfectly stable, there would be no mutations, no evolution, no change. Evolution selected for the right amount of stability. The Goldilocks zone. Enough to preserve information across generations. Enough to maintain the identity of species, but not so much that change became impossible. Just enough imperfection to allow life to adapt. Just enough stability to allow life to continue. The cells in your body have multiple mechanisms for protecting and preserving your DNA. Nucleotides, the building blocks of DNA, are among the most carefully protected molecules in the cell. The DNA sits protected in the nucleus, surrounded by a nuclear membrane. The nucleus has pores, but those pores are selective. What can enter and exit is carefully controlled. The DNA is wrapped around histones, protein spools that protect it from damage and control access. The histones themselves are protected from degradation more carefully than most other proteins. The cells have dedicated machinery for repairing DNA damage, photolyase enzymes that fix UV damage, nucleotide excision repair systems that remove damaged sections, mismatch repair systems that catch errors before they become permanent, double-strand break repair mechanisms that fix breaks in the DNA backbone. The cell invests enormous energy in preserving the DNA, because the DNA is the blueprint. Damage the blueprint and everything falls apart. And so DNA persists. It persists in the changing cells of your body. New cells are being born constantly, each one a copy of the original, carrying the original DNA. It persists across the generations of your family. DNA that was in your parent is now in you. DNA that will be in your children. It persists in your species. Every human carries DNA that connects to every other human. It persists across millions of species on Earth. It persists in the tiniest microbes in the deepest parts of the ocean. It persists in the plants and animals we share the world with. It persists in your gut bacteria, in the soil microbes, in the trees in the forest, in the fungi that break down dead wood, in the insects that pollinate flowers. DNA is everywhere life exists. And it has been everywhere life has existed for over 3 billion years. The molecule that stores genetic information has proven so effective that almost every organism that has ever evolved has used it. The only competitors were RNA-based organisms billions of years ago, before DNA took over, before DNA proved itself so superior that it became the standard system for storing genetic information across the living world. This persistence is not dramatic. It is not flashy. It is quiet, steady, relentless, invisible. DNA just keeps doing what it does, replicating with stunning accuracy, getting damaged occasionally, being repaired usually, mutating rarely, being passed on constantly, accumulating changes slowly, creating diversity gradually, allowing evolution to happen, enabling life to persist and adapt and explore every possible ecological niche on the planet. It is not animated by any will or purpose. It is just chemistry. Molecules responding to other molecules according to the laws of physics and chemistry. No consciousness, no intention, no awareness, just chemistry happening. Blind, purposeless chemistry. And yet, through that chemistry, through that blind, purposeless process, DNA has created the most complex, diverse, intricate systems the universe has yet produced. Life in all its abundance, life in all its forms, life that thinks, feels, creates, imagines, loves, all of it built from the persistence of a molecule, all of it emerging from chemistry. Your DNA is not special because it is unique. Most of it is not unique. Most of your DNA is virtually identical to your neighbor's DNA. But your DNA is special because it is yours. It carries your personal instructions. It makes you, you. And yet, those personal instructions are built from a shared system, a shared language, a shared history, a shared process that goes back to the origin of life. You are an expression of universal principles applied specifically to your situation. You are a unique instance of a universal process. You are personal and ancient and connected and individual all at once. This is the paradox of DNA. It is both profoundly personal and profoundly universal. It is yours alone, in its specific sequence, in its specific combination. Yet it is shared with every other person on Earth, shared with apes and mice and fish and plants, shared with the bacteria in your gut, shared with all life, the same system expressing itself in infinite variations, the same language telling infinite stories. The library is yours, and it belongs to all of us. But there is more to DNA than just genes. For decades, scientists thought they understood DNA completely. They thought DNA was just a list of genes. Genes that encoded proteins. Proteins that did the work of the cell. Everything else was junk. Everything else was noise. Evolutionary leftovers with no function. Garbage DNA that the cell carried around for no particular reason. But science changes. New technologies allow us to see things we could not see before. And what scientists discovered was that DNA is far more complex than they realized, far more sophisticated, far more interconnected with everything else in the cell. Less than 2% of human DNA codes for proteins. Less than 2%. This means that more than 98% of your DNA is not directly making proteins. For a long time, that meant scientists thought 98% of your DNA was useless. But that interpretation was wrong. That 98% includes regulatory regions, control sequences, switches that turn genes on and off, enhancers that amplify gene expression, silencers that suppress it, promoters that mark where genes begin, terminators that mark where they end, repetitive elements that might have once been viral sequences but have become integrated into the genome, structural DNA that helps organize chromosomes, spacer sequences that separate genes from each other. This non-coding DNA is not junk. It is control panel. It is the operating system. It is the management layer that determines how and when the genes actually function. And there is more. There are sequences that produce RNA molecules that are not translated into protein. RNA molecules that have their own functions, that regulate other genes, that participate in splicing and translation, that help organize the cell, that participate in immune responses. These are functional sequences. They are not genes in the sense of protein coding genes, but they are genes in the sense that they are units of functional DNA. Regions of DNA that have a specific role, a specific purpose. and they are essential to life. The discovery that DNA contains this vast landscape of regulatory and functional sequences has revolutionized biology. It has revealed that the cell is not simply executing a list of protein instructions. The cell is a complex system where genes are constantly being turned on and off based on signals from the environment, from other cells, from the organism itself. The DNA is the blueprint, yes, but the blueprint is far more complex and dynamic than anyone realized. The blueprint contains not just static instructions, but also the mechanisms for controlling when those instructions are read and how intensely they are followed. This is the realm of epigenetics, a field that studies how DNA is controlled and regulated without changing the underlying DNA sequence itself. Epigenetics explains how two people with virtually identical DNA can develop as different organisms based on different environmental exposures. It explains how identical twins can become increasingly different as they age and experience different lives. It explains how a cell in your liver that contains the same DNA as a neuron in your brain can develop into a completely different type of cell with completely different functions. The DNA sequence is identical. The genes are the same, but the expression of those genes is different, completely different. And that difference is coded in the regulatory layers that sit on top of the DNA sequence. In the epigenetic marks that tell the cell which genes to read and which to ignore, this understanding has transformed how scientists think about DNA. It is no longer just a static code. It is a dynamic system, a responsive system, a system that can be influenced by the environment, a system that can change during your lifetime in response to how you live. The DNA sequence itself remains constant, but the expression of that sequence is malleable, changeable, influenced by choice and circumstance. This is where the metaphor of DNA as a library becomes even more apt. A library is not just a collection of books. A library is also a system of organization, a system of access, a system that determines which books are available, which are in frequent circulation, which are archived and hard to access. DNA is the same. The genetic sequence is the collection of books. The epigenetic marks are the organizational system that determines how those books are used. And that organizational system is not fixed. It changes. It responds to the environment. It responds to experience. It responds to food, to stress, to toxins, to exercise, to emotional state, to sleep patterns, to relationships, to meaning, to every aspect of how you live. Your environment is writing on top of your genetic code, not hanging the code itself, but changing how the code is expressed, changing which genes are active, changing how intensely they are expressed. Your life is literally being written into the regulatory layers of your DNA. You are a book that is constantly being rewritten. The text stays the same, but the interpretation changes. The emphasis changes. What is highlighted and what is left in the background changes. You are a dynamic system built on top of a static foundation. This is the profound insight of epigenetics. You are not just your genes. You are your genes in the context of your life. Your genes plus your environment equals your phenotype, the physical expression of what you are. And that expression is not fixed, not determined at birth. It is dynamic. It changes. The same DNA sequence can produce different outcomes depending on what is happening around that DNA, depending on what signals the cell is receiving, depending on what your body needs. Consider the work of researchers studying Dutch children who were in utero during the Dutch famine of 1944 and 1945. These children had limited nutrition in the womb. Their mothers were starving, and yet these children survived. They were born. They grew. But something had changed in their DNA, not the sequence, but the methylation pattern, Epigenetic marks had been laid down that would influence their metabolism for the rest of their lives. These children, as adults, had higher rates of metabolic disease, higher rates of obesity, higher rates of diabetes. The deprivation they experienced in the womb had written itself into their epigenetic code. And that code was influencing how their body processed food for decades after the famine had ended. The DNA sequence was the same as their sibling, GS, who were not in the womb during the famine, but the epigenetic marks were different, and those marks had consequences. Or consider identical twins, twins who share the exact same DNA sequence, born at the same time, raised in the same house by the same parents, and yet, as they age, they become increasingly different. Their epigenetic marks diverge. Some genes that are active in one twin become inactive in the other. Some genes that are quiet in one become active in the other. They experience different lives, eat different foods, exercise differently, experience different stresses, and all of those differences get written into their epigenetic code, the same DNA, differentiated by experience, the same blueprint, interpreted differently based on environment and circumstance. After 30 years, identical twins can look quite different, have different health outcomes, develop different diseases, be different people in many meaningful ways, the same genes, different epigenetics, different outcomes. This is why DNA is not destiny. Yes, you inherit your genes from your parents. Yes, those genes constrain what you can be. Someone born with genes for short stature will not become a basketball player no matter what. Someone born with genes for blue eyes will not develop brown eyes through willpower. There are limits set by genetics. But within those limits, there is freedom, enormous freedom. The expression of your genes is not fixed. It is responsive. It is changeable. The choices you make, the food you eat, the stress you experience, the exercise you do, the love you feel, all of these are being registered in your cells. All of these are influencing which of your genes are expressed and which are silenced. All of these are writing themselves into your epigenetic code. You are not just reading the instruction manual of your DNA. You are writing notes in the margins. This is genuinely calming when you think about it, because it means you are not locked into what your genes say. You are not a prisoner of your genetic code. You are a participant in how that code is expressed. You are an active agent in determining which of your genes get expressed. Your lifestyle is the pen. Your choices are the ink. And the page is your epigenetic code. You are writing your own biological story, even as you are bound by the story written by your ancestors in the sequence of your DNA. You are both constrained and free, both determined and determining. This is perhaps the most empowering truth that molecular biology has uncovered. You have influence over your own genome, not over the sequence, but over the expression. And in a very real way, that matters more. And there is more to this story. Because the epigenetic marks you create do not just affect you. Some research suggests that epigenetic marks can be inherited. That marks created in response to your environment might be passed on to your children. Not genetic inheritance, because the DNA sequence is not changed, but epigenetic inheritance. The marks that modify how your genes are expressed might be carried forward to the next generation. Your life experience might influence not just your biology, but potentially the biology of your children. This is still an emerging area of research. Scientists are still figuring out the mechanisms and the extent to which this happens. But the possibility is profound. Your life might be writing itself not just into your epigenetic code, but into the epigenetic code of your children. Your great-grandchildren might be living with epigenetic marks that were created in response to your life. The past reaches forward. Your choices echo through generations. You are not just living your own life. You might be writing the epigenetic code of your descendants. This is the true power of DNA. It is not just a passive storage system. It is an active participant in your biology. It is responsive. It is influenced by how you live. It is shaped by your choices. And through it, you can influence not just your own biology, but potentially the biology of those who come after you. You are an author as much as you are a reader of your own genetic code. The DNA in your cells is ancient. It carries the history of billions of years, but it is also completely contemporary. It is responding to what you had for breakfast, to your stress levels, to your exercise, to your relationships, to every choice you make. The past and the present are woven together in the structure of DNA. History and possibility, constraint and freedom, inheritance and choice, all of it is encoded in that double helix. All of it is you. And this understanding changes everything about how we think about health and disease. If DNA is not just your genes, but your genes in the context of your life, then disease is not simply genetic destiny. Yes, some diseases are strongly influenced by genetics. If you inherit mutations associated with certain cancers, your risk is elevated. If you inherit mutations for Huntington's disease, you will develop the disease. There are genetic constraints. But even in those cases, the expression of disease might be influenced by epigenetics. Stress can activate cancer genes. Environmental exposures can change which genes are expressed. The exact timing and severity of disease can be influenced by factors outside the genetic code. And for many of the chronic diseases that afflict modern society, DNA is just one factor among many. Diabetes, heart disease, Alzheimer's, obesity. These diseases are influenced by genetics, but they are shaped far more powerfully by how you live, what you eat, whether you exercise, how much you sleep, your stress levels, your relationships, your sense of purpose. These factors are writing themselves into your epigenetic code every single day. They are determining which of your genes get expressed. Some people with genetic risk factors for these diseases live long, healthy lives because they live in ways that silence those disease genes. Other people without genetic risk factors develop disease because they live in ways that activate genes that would otherwise stay quiet. The DNA is the piano. The lifestyle is the pianist. The music that emerges depends on both. This is why understanding DNA is not just about understanding genetics. It is about understanding how to live, how to make choices that support the expression of genes that promote health and suppress the expression of genes that promote disease. It is about understanding that you have influence over your biology. Not complete control, but real influence. You are not a prisoner of your genes. You are a collaborator with them, working together, your choices and your genes in conversation, each influencing the other, each shaping the outcome. The DNA in your cells is a record of everything. Everything you eat, everything you experience, every stress you endure, every moment of joy, every relationship, every loss, all of it is being written into your epigenetic code. Your DNA is not just a blueprint, it is a journal, a diary of your life written in the language of gene expression. And the remarkable thing is that the journal is not closed. You are still writing. Every day, every choice, every moment, you are writing yourself into your DNA. You are becoming who you are through a million tiny interactions between your genes and your life. And that process will continue until you die. You are never finished. You are never locked in. You are always becoming, always writing, always expressing yourself through the ancient machinery of DNA that has been passed down through billions of years and millions of ancestors. You are the latest chapter in a story that began when life first emerged on Earth, and you are still writing. But here is where the story gets even stranger. For a long time, scientists looked at DNA and made a profound mistake. They saw that less than 2% of it coded for proteins. Less than 2%. And they assumed the rest was garbage. Evolutionary leftovers. Junk DNA was the actual term they used. Junk. Not mysterious DNA. Not unknown DNA. Junk. As if somewhere in the depths of the cell there was just waste material being carried along, taking up space, serving no purpose, waiting to be eliminated. Imagine discovering a library and deciding that 98% of it was trash because you could only understand the 2% that was filed in the fiction section. Everything else, all the reference materials, all the catalogues, all the organizational systems, all the historica, records, all the indices and guides and cross-references, all of it labelled as useless junk taking up valuable space. This is how scientists approached DNA for decades, with enormous confidence in their misunderstanding. That was the mistake, because it turns out that those regulatory regions, those sequences that weren't directly making proteins, they were doing something far more important. They were controlling which proteins got made and when. They were the switches, the dimmer controls, the on and off buttons for the entire system. Those sequences that scientists thought were evolutionary garbage were actually the operating system, the control panel, the management layer that determines how the body actually functions. And this discovery changed everything about how we understand DNA. The non-coding DNA contains enhancers that amplify gene expression, turning genes louder or quieter as needed, silencers that suppress it, muting genes that shouldn't be active in particular cells, promoters that mark where genes begin, providing entry points for the molecular machinery, terminators that mark where they end, telling the cell when to stop reading. These aren't randomly scattered remnants left over from evolution. They're precisely positioned regulatory elements that control the timing, location, and intensity of gene expression. A single enhancer might sit thousands of base pairs away from a gene, yet still influence exactly how much protein that gene produces. Not a little, not approximately, exactly. The cell has evolved mechanisms to recognize these distant sequences and respond to them instantaneously, bending the DNA into loops so that the enhancer is physically close to the gene, even though they're far apart in the sequence. It's like having a remote control that can affect a television in another room that you can't even see, yet the effect is immediate and precise. The signal travels through the DNA, like electricity through wires. The discovery of regulatory DNA was not sudden. It was gradual, painfully gradual. Technology improved. Scientists could look at DNA more carefully. They could sequence it. They could read the letters and find the genes. But they could also compare sequences from different species and see which regions changed and which stayed the same. They could track gene expression patterns. They could see which genes were active in which cells and which were silent. They could watch how patterns changed in response to signals. They started noticing something odd about those non-coding sequences. They were highly conserved across species. If they were truly junk, they would have accumulated mutations and changed drastically over millions of years of evolution. Instead, they were preserved, carefully maintained, passed down through evolution with minimal changes. That kind of conservation only happens when something matters, when it provides a survival advantage, when organisms that have it thrive and their offspring inherit it, and organisms that lose it through mutations struggle or die without reproducing. Evolution doesn't preserve things that don't matter. Evolution preserves things that work. So scientists began to revise their understanding. The junk DNA wasn't junk. It was regulation. It was control. It was the system that determined how the genetic code was actually expressed in a living organism. And this revelation opened up entirely new fields of biology. Epigenetics became a real science with real mechanisms. Gene regulation became understood as something complex and sophisticated and integral to all of biology. And people began to realize that if you wanted to understand life, you couldn't just look at the genetic sequence. You had to look at how that sequence was controlled and expressed and modified by environmental factors and cellular signals and developmental timing and experience. The DNA itself is static. The expression of the DNA is dynamic. That distinction changed everything. This realization is also profoundly relaxing in its own way, because it means that your DNA is not just a list of predetermined instructions handed down from your ancestors with no possibility of modification. It's a flexible system, a responsive system, a system that can be influenced and modified and controlled by signals from your environment. The regulatory DNA that scientists once called junk is the part that makes you adaptable, the part that allows your body to respond to change, the part that lets you grow and develop and adapt throughout your lifetime in response to challenges and opportunities. The genetic code itself remains constant. The sequence of base pairs doesn't change, but the way it's expressed is dynamic, responsive, influenced by everything you do and experience and encounter. Your DNA is not a rigid instruction set. It's an instruction set with annotations and margins and commentary that can be written and rewritten throughout your life. The same genes can produce different results based on how they're regulated. The same genetic code can express itself in different ways depending on circumstances. This flexibility is what makes life possible. This is what makes adaptation possible. This is what makes you capable of changing in response to your world. Today, DNA has become ubiquitous in ways that would have seemed like science fiction just a few decades ago. Your DNA can be used to identify you more reliably than a fingerprint, more reliably than facial recognition, more reliably than any other method humans have ever developed. A single strand of hair left at a crime scene, shed 3 months ago. A drop of blood on a defendant's jacket, dried and ancient. A flake of skin under a victim's fingernail. DNA. Evidence has revolutionized forensic science completely. It has exonerated innocent people who spent decades in prison for crimes they did not commit. Innocent people who would have been executed if DNA technology had not existed. It has identified guilty people who thought they were untouchable, who thought they had committed the perfect crime. It has created a permanent record that cannot be argued with or denied or reinterpreted based on perspective or bias. The molecular structure of DNA, the way it replicates and the way it codes for traits, makes it perfect for identification. Every person on Earth has a unique combination of genetic variations, not in the basic sequence. The basic sequence is 99.9% identical B, as in all humans, but in the variations scattered throughout. The small differences, the mutations that accumulated in your family line over generations and are unique to you and your relatives. These markers are scattered throughout the genome like breadcrumbs. They identify you with almost perfect certainty. A match of just 13 genetic markers makes it virtually certain that a piece of DNA belongs to a specific person. The odds of someone else having the exact same 13 markers are in the billions. This has profound implications for justice. For the first time in human history, guilt or innocence can be determined at the molecular level, not based on testimony that can be false or mistaken, not based on circumstantial evidence that can be misleading, not based on eyewitness accounts that can be influenced by bias and memory error, but based on the actual physical evidence of who was present at a scene, based on molecules that cannot lie because they have no bias. And while this has obvious applications in law enforcement, it's also profoundly unsettling in other ways, because it means that your genetic information is data. It can be stored, analyzed, compared against databases, shared between agencies. It can be used to track you, to predict your future, to identify your relatives without their consent. People have discovered family members they never knew existed, have been forced to confront unexpected biological relationships, have had their genetic information used to investigate crimes they had no connection to. The same technology that solves crimes can also be used for surveillance, for genetic profiling, for discrimination, for control. DNA has become a double-edged sword, incredibly useful, but also potentially incredibly dangerous if misused. At the same time, DNA technology has revolutionized medicine in ways that are still unfolding. Genetic testing can now identify whether you carry mutations associated with certain diseases. Tests for BRCA mutations that increase breast cancer risk. Tests for familial hypercholesterolemia that dramatically increases heart disease risk by disrupting cholesterol metabolism. Tests for Huntington's disease mutations that guarantee you'll develop the disease if you live long enough, that turn healthy neurons into failing ones years or decades in the future. These tests give people information about their future. Precise information. Not probabilities or risk factors. Certainties. And that information can be used to make better choices, to get screened more frequently, to make lifestyle changes that might slow disease progression, to take preventive medications, to prepare emotionally and financially and psychologically for what might come. Or it can be ignored, or it can be devastating. This is powerful. This is valuable. It's also sometimes profoundly difficult. Knowing that you carry a mutation that guarantees you'll develop a serious disease is not something most people are prepared for emotionally. It changes how you see your future. It changes your life whether you want it to or not. The promise of personalized medicine is that doctors will eventually be able to look at your unique genetic makeup and prescribe treatments tailored specifically to you, not the average treatment that works for most people. Your treatment, based on your DNA, based on how your unique genetic variations respond to different drugs and environmental factors. The same medication that works wonderfully for one person might have terrible side effects in another person with different genetic variations in drug metabolism genes. Someone might process a medication so quickly that it never reaches therapeutic levels. Someone else might process it so slowly that it accumulates to toxic levels. The same dose that heals one person could kill another. Personalized medicine promises to eliminate this one-size-fits-all approach, to match treatments to individuals, to make medicine more effective and safer. We're not there yet. The science is still developing. We're still learning which genes matter for which drugs. We're still building the databases of genetic variations and drug responses, but the direction is clear. DNA is going to become increasingly central to medical decision-making. It will become standard to have your genome sequenced before starting a new medication. It will become routine to adjust doses based on your genetic variants. And then there's the most surprising application. DNA is being used as a data storage medium, not for biological information, but for human information, for culture, for knowledge, for history. In 2013, researchers at the European Bioinformatics Institute encoded Shakespeare's sonnets into DNA, all 154 of them. They also encoded a photograph, a color photograph. Not simple, Not easy. They also encoded an audio clip from Martin Luther King's "I Have a Dream" speech, one of the most important speeches in human history. They encoded Watson and Crick's original research paper describing the DNA structure, the paper that changed science forever, and they encoded detailed instructions on how to decode it all, how to extract the meaning from the molecules. They synthesized the DNA, they stored it physically, and then they sequenced it using standard DNA sequencing technology. And they recovered all the original information perfectly, with 100% accuracy, no errors, no corruption. Every single bit of data was intact, every word of Shakespeare exactly where it should be. The audio clip perfectly reconstructed. It worked. Think about this carefully. Shakespeare's words from the 16th century encoded into a 21st century molecule, preserved perfectly. Not on a hard drive that could crash if the power fails or if the magnetic surface corrodes. Not on a tape that could degrade as the polymers decompose. Not in a cloud that could be hacked or could vanish if the company goes bankrupt, but in molecules of DNA. Molecules that can last for thousands of years if stored properly. Maybe tens of thousands of years. Maybe much longer. Molecules that are so incredibly compact that the entire world's information, all the data that existed in 2012, could theoretically be stored in about 4 grams of DNA. 4 grams. Less than the weight of a teaspoon. Imagine all the books ever written, all the music ever recorded, all the films ever made, all the conversations ever had, all the code ever written, all of human culture and knowledge and history, compressed, encoded, stored as molecules, shrunk down to a few grams. DNA is the ultimate storage medium, more durable than anything humans have invented, more compact, more elegant, more perfect. This raises genuinely fascinating questions about the future. What if we stored the most important information about human civilization in DNA? What if we took the collected wisdom of humanity and encoded it into molecules and buried it in time capsules? What if we left it for whatever comes next? Aliens finding it billions of years in the future? Humans living in the far future, living on Mars or the moons of Jupiter? Surviving some catastrophe that kills most of civilization, but leaves behind DNA storage vaults? It would be evidence of what we were, what we created, what we valued. Your genome is storing biological information that makes you alive. But DNA could also store human culture, human knowledge, human history, art, music, literature, philosophy, science. Everything worth remembering. Ring could be written into DNA and preserved indefinitely. It's a profound thought. The molecule that carries your genetic instructions could also carry the entire legacy of human civilization. The code of life itself could become the storage medium for human meaning. Your DNA is unique, genuinely unique. Of the 3 billion base pairs in your genome, only about 15 to 20 million of them vary between you and the next person. That's less than 1%. That's 0.6% more precisely. But that minuscule, less than 1% creates the differences between you and everyone else on Earth. It creates your eye color, your hair color, your height, your metabolism, your predisposition to certain diseases, your intelligence, your personality, your talents and abilities and weaknesses. Everything about you that is genetically influenced comes down to those tiny variations scattered throughout your genome. A single base pair change, just one letter of the genetic code different, in one gene. That can dramatically affect a protein's function, can change how your body processes food, can change how your immune system works, can change your risk of developing disease, can change whether you can taste certain flavors or smell certain scents. A single base pair. Out of 3 billion, That's how precise and powerful genetic variation is. 1 letter out of 3 billion changed, and your life is different. And here's the remarkable thing that really puts everything in perspective. Most people on Earth are more genetically similar to each other than they are different. Humans are 99.9% genetically identical. Think about that. You and a stranger on the other side of the world share 99.9% of your DNA. The genetic differences between a person from Japan and a person from Brazil are smaller than the genetic differences within African populations. Africa is the most genetically diverse continent because all of humanity originated there thousands of years ago and then spread out to populate the rest of the world. As people migrated to other continents, they carried only a subset of the genetic variation that existed in Africa. So African populations, the populations that never left, retained more genetic diversity than any other part of the world. This means that the common genetic categories that people use to divide humanity don't really match genetic reality at all. The genetic differences don't align neatly with racial categories or ethnic boundaries or national origins. Genetics cuts across those divisions. Genetics doesn't recognize the categories that humans have created. We're all related. We're all cousins. We're all more similar than we are different. The variation that makes some of us look different from others is superficial compared to what we share. Our skin color, our hair texture, our eye shape. These visible differences come from a tiny fraction of our genetic code. The vast majority of our genome is identical, but the differences that do exist are meaningful. They're written in your DNA right now. They make you you, a specific combination of genetic variants that probably doesn't exist anywhere else on Earth. A unique edition of the human genome, a personal library containing the same books as everyone else, but with unique bookmarks in unique places. Billions of people carry similar genomes because we all carry the same basic genes. We all have the same general structure, the same basic biochemistry, the same essential processes running in our cells. Humans are more similar than we like to admit, but the minor variations in those genes, the SNPs, single nucleotide polymorphisms, the places where one person's DNA differs by a single base pair from another person's DNA. Those variations scattered throughout your genome. Those are what make you unique. They make you distinguishable. They make you specifically you and not anyone else. This is how we can identify people with DNA, not because the DNA is completely different from everyone else's, but because the specific pattern of variations is unique. Like a barcode written in molecules, like a fingerprint written in the fundamental code of life. Every cell in your body carries this barcode. It's in your blood, your hair, your skin, your saliva, your sweat, your tears. Leave a trace anywhere and it could potentially be used to identify you or rule you out as a suspect. This is amazing for justice. It's also unsettling for privacy. Your genetic information is essentially a perfect identifier. You can't hide from it. You can't disguise it. You can't claim to be someone else if the DNA says otherwise. Your genome is your signature written in molecules. Before we talk about those ethical questions, there's something else you should know. Your DNA is not the only DNA that matters to your biology. Your body isn't just you, it's an ecosystem. Trillions of bacteria live inside you, in your gut, on your skin, in your mouth, throughout your body. About the same number of bacterial cells as human cells, about 37 trillion of each. These bacteria have their own DNA, their own genomes, millions of genes that are not yours, but that affect your biology constantly. They help you digest food. They produce vitamins your body can't make on its own. They interact with your immune system. They protect you from harmful pathogens. They influence your mood through neurotransmitters. You cannot survive without them. You are not an individual organism. You are a collective. You are a holobiont, a human, plus the microbiota living with you. Your health is their health. Their health is your health. Your genome is inseparable from their genomes. When scientists study human genetics, they're increasingly realizing they can't understand human biology in isolation. You have to understand the microbiome. You have to understand the ecosystem of organisms living in and on your body. You are never truly alone because you were never meant to be alone. You were designed through evolution to be a community, and the DNA of other organisms surrounds you constantly. Researchers vacuumed up air from zoos and sequenced all the DNA they found. DNA from birds and mammals living in the zoo. DNA from wild animals hundreds of meters away. DNA from the food given to the zoo animals. All of it just floating in the air. You are breathing in the genetic material of thousands of organisms every single moment. Fish, DNA. Bird, DNA. Plant, DNA. Bacterial, DNA. Viral, DNA. All of it floating in the atmosphere around you. The world is full of DNA. It's in the soil, in the water, on every surface, in the air. You cannot escape it because it's everywhere life has ever been. You are not just living among other organisms. You are living in a cloud of genetic material, in a swirling dance of DNA that belongs to every living thing around you. The world is not separate from you. You are breathing it. You are part of it, and it's part of you. And this raises profound ethical questions that we are just beginning to grapple with. Who should have access to your genetic information? Your government, your employers, your insurance companies, your parents if you're a child. Should genetic information be used to deny you insurance because you carry genes associated with disease? Should it be used to deny you jobs because of genetic predispositions? Should it be used by the state to build comprehensive genetic databases of everyone in the population? Should your genetic information be shared with relatives without their consent? Should pharmaceutical companies be allowed to access your genetic data to develop drugs? These questions don't have easy answers. The technology is powerful and neutral. It can be used for tremendous good, identifying criminals, preventing disease, curing inherited disorders, finding biological relatives. But it can also be used for tremendous harm, genetic discrimination, surveillance, control, creating permanent genetic records that could be misused by future governments with different values. The molecule that has preserved humanity's genetic heritage since the dawn of life is now in human hands. And we're still figuring out what we should do with it. We're still debating the ethics. We're still arguing about privacy and access and ownership. These are questions that will define our relationship with DNA for generations to come. Your DNA is ancient. It's also completely contemporary. It's also the future unfolding in the present moment. The mutations that created your unique genetic variation happened in your parents, in your grandparents, in your ancestors, stretching back through time. All those tiny changes accumulating, all those variations becoming part of you. And the DNA in your cells right now, the mutations that you carry, some of them will be passed on to your children if you have them. They will become part of the future genetic landscape. Every person who reads this and has children and passes on their DNA is participating in evolution, is creating new combinations of genetic variation, is shaping what humanity looks like in the next generation and the ones beyond. Natural selection is no longer the primary force shaping human evolution in the way it once was. Medical technology extends the lives of people who might have died young, allowing them to reproduce. Cultural values and personal choice determine whether someone has children and how many. Humans are selecting mates based on attraction and compatibility, not fitness for survival. But evolution is still happening. Your DNA is still mutating. Humans are still changing. We're just changing in different ways now. The pressures are different. The mechanisms are different. but the fundamental process continues. This is where DNA becomes not just a molecule, but a bridge between past and future, between the ancient history of life itself and the unwritten future of humanity that has not yet been written. You carry in your cells the genetic legacy of everyone who came before you. Thousands of years of ancestors, hundreds of thousands of years if you trace back far enough, millions of years if you look at all of humanity's primate ancestors. And if you have children, you will pass on that legacy to them. You will also pass on the new mutations that occurred in your lifetime, the variations that make your genetic line unique. You will contribute to the genetic diversity of the human species in this moment. You will be a single link in the chain that stretches back to the origin of life billions of years ago and forward into a future you will never see. DNA remembers everything if you know how to read it. It remembers the evolution that led to you, the countless adaptations that accumulated over billions of years of trial and error and survival, the changes that turned fish into amphibians, amphibians into reptiles, reptiles into mammals, mammals into primates, primates into apes, apes into humans. Every single step of that journey left molecular signatures in your DNA, evidence of where we came from. You can read that history in your cells. You can see it in the genes you share with fish that your distant ancestors were. The genes you share with mice that help us understand how your brain works. The genes you share with plants that help you convert sunlight into energy in the mitochondria of your cells. The deeper you look, the more connected everything becomes. The more obvious it is that all life shares common ancestry. All life uses the same genetic code. All life relies on DNA to pass information from one generation to the next. All life emerged from a common ancestor that lived billions of years ago and has been evolving ever since. And yet that common code, that shared genetic foundation, creates infinite diversity. The same molecular machinery in a bacterium and a blue whale. The same basic principles of replication and mutation and selection. But in a bacterium, it creates a single cell that can survive in boiling water or freezing acid or the vacuum of space. In a whale, it creates an organism that weighs 100 tons and can hold its breath for 30 minutes and navigate the oceans with sonar so sophisticated that it bounces sound off molecules and calculates distance with extraordinary precision. In a human, it creates a brain capable of writing poetry about itself, capable of building computers that can simulate thought, capable of contemplating its own existence and wondering what it all means. The same code, different expressions. The infinite variation, created by 3 billion base pairs of information being read and interpreted and expressed in countless different ways across the tree of life. This is what makes DNA so calming when you really understand it. Because it connects you to everything. It connects you to your ancestors stretching back through time to the origin of life. It connects you to your descendants who have not yet been born. It connects you to other humans across the world. It connects you to animals and plants and fungi and bacteria. It connects you to all life on Earth. You are not separate from nature. You are nature. Your body is made of the same molecules, controlled by the same principles, operating according to the same laws of physics and chemistry that govern all living things. And those laws have been tested by billions of years of evolution, refined by countless mutations and selections and adaptations, perfected through natural selection into systems of breathtaking elegance and efficiency. The DNA in your cells is the product of the most intense quality control process ever conceived. Every living organism alive today is the descendant of organisms that survived, that successfully reproduced, that passed on working genes to their offspring. You are here because your ancestors' DNA worked, because it was good enough, because it allowed them to survive long enough to have children. That's an incredible thought. You exist because your DNA works. Because it has been working in various forms for 3.5 billion years. And it will continue to work, modified and changed and mutated in ways we cannot predict. In your descendants for as long as life exists on Earth, as long as the planet itself exists, as long as the sun continues to shine. You are a temporary expression of something ancient and profound and ongoing. You are a note in a song that has been playing since the beginning of life and will continue long after you are gone.